Yes, so today I will share the stage with Ashley. He will cover a bit concepts behind brain life. And hopefully today we will make reproducible neuroimaging easier for you by providing a practical guide to software containers. This is the one. We have no financial interest whatsoever to disclose, although both of us are part of the Neurodesk team and Ashley is also part of the Brain Life team. So let's start with a safe assumption. I would say that most of you have some sort of some computational component to your research. And if you're here, it is because you care about open science and reproducibility. So I wonder how many of you have struggled to install neuroimaging software on your local computer? Please raise your hands. Well, yeah, that's a couple. And if you struggle to install this software on your local computer, you might think that you would never port your analysis to HPC clusters. But hey, let's say that installing this standard neuroimaging software wasn't that bad. What about more specialized tools, like something developed by a collaborator or a lab mate? How many of you have struggled to recycle these tools from your collaborators? So I would say it's a bit worse, right? And I would imagine it wouldn't be without their help. Well, if it is any consolation, you are not alone. This is not just a neuroimaging problem. It's a challenge across all domains that leverage scientific software. But I'm not here to judge, especially because I was not trained to be a computer scientist, and I was not taught to do better. But I would recommend you guys to check out the new book Russ Poljarek is posting on Substack. It's quite good. So I'm actually here to suggest that software containers may be the solution to this problem and many other problems. So our goal today is that you leave this session knowing what a software container is, the benefits of using software containers, and how you can leverage these software containers via platforms like Neurodesk and BrainLife. But before we get into the nitty-gritty, I'd like to motivate you to use software containers as soon as OHBM is done. And I will motivate you by sharing some challenges that we face with scientific software. We can classify these challenges into 5 categories: portability, compatibility, computational reproducibility, scalability, and project management. And if you want to know more about these different concepts, you can check out this nice Nature Reviews paper. Portability and compatibility are intertwined concepts which are closely related to the fact that scientific software is often not self-sufficient. It requires system tools, different libraries, which we call dependencies. And that means that it is not really straightforward to install the same software version on different machines. In practice, you always have to install a software version that is compatible with your specific operating system. And because you do that, and the system dependencies vary, you might run into the problem of computational reproducibility. Researchers have found that results may differ even when you are using the same data and the same software, which is caused by variations in the underlying software dependencies. So if this is not a major problem in itself for you, there is more. Because you have to install software compatible with your operating system, you always have to do extra work if you want to port your analysis to a HPC cluster or cloud resources, which means that scaling up data processing is not necessarily straightforward. And needless to say that to install other softwares that require different dependencies, or better saying, dependencies that have different versions may lead to bad behavior of installed software. So, for example, here we could have 2 different pieces of software that require the same dependency but different version. And when you install these two pieces of software on your computer, they will behave poorly. But what if we made software somehow isolated, such that there wouldn't be any conflict between dependencies? Well, this is essentially what software containers are. A software container is a virtual package that encapsulates all information needed to run computer code, including system tools, library settings, and the application itself, as well as any operating system components that are not provided by the host operating system. In other words, it is a technology that allows us to package our software and dependencies into an isolated standard unit of software that is completely self-contained. And because they are self-contained, they can be run in any operating system so long the computer has the container runtime installed. And because a container includes all the dependencies to run a specific software, and they are lightweight, you can also port them to any computer, just put in a USB stick. For the same reason, they also enable computational reproducibility, because the same container can be run in any operating system. Software containers are also portable to any computational environment, which means you can leverage them in HPC clusters and cloud resources. Finally, because they are completely isolated, you can have multiple of these containers and combine them like Lego pieces to run different pipelines and different projects. In summary, there are several benefits to software containers. They are portable, usually less than 10 gigabytes. They are compatible to any operating system. They enable computational reproducibility. They enable scalability, as they are compatible to multiple computational environments. And they facilitate project management, as they are self-sufficient and isolated units of software. Well, now that we know what software containers are and how we can benefit from using them, we can get to the fun part, which is learning how we can use them. First, you may consider building containers using some containerization platform. And they provide a standard format for packaging and distributing applications. And they include tools for creating, managing, and deploying containers. Amongst the most common containerization platforms, we have Docker and Singularity. Today, we can use Docker as our working example, but just so you know, Singularity is usually preferred in HPC cluster for security reasons. To build and run a Docker container, we start by writing our recipe with all the commands required for installing software and dependencies and downloading any required file. This recipe is what we call Docker file. And then we are ready to build our Docker image, which is essentially a read-only blueprint of our container. And finally, we are ready to run our Docker image, and that generates a Docker image instance, which is our Docker container. So in other words, a Docker container is a runtime environment. So let's have a look at how a Docker file looks like. Here we have an example from FreeSurfer, where we start by defining a base image. So here we have CentOS, but you could have Linux, Ubuntu, any operating system, or actually you could use any pre-built Docker image as your base image. Then we have some run commands to install any dependencies and the required software, which is shown in the middle, line 24. And finally, you can configure your environment variables as shown here using the env commands, multiple lines down there. From a Docker file, you can build your Docker image by simply running docker build and passing the name of your Docker image and the path to the Docker file. Once your Docker image is built, you can share your Docker image on Docker Hub, so anyone can pull it and use on their own research. And this is simply done by first tagging your Docker image with your Docker Hub username and then pushing it to Docker Hub. Quite simple. But an easier alternative to writing your recipe and building your Docker image is to simply pull a pre-built image from Docker Hub, where you can find many working images. And you can do so by running Docker pull. Finally, once you have your Docker image, you're ready to run it using Docker run, and you will have your Docker container. But the remaining question is, do we need to do all of that to leverage software containers in our work? Well, I'm here to say that no, we don't have to do all of that work because we have neuroimaging data analysis platform that allow us to do it without the burden of managing multiple software containers. Today we discuss NeuroDesk and BrainLife. So NeuroDesk is an open, reproducible, scalable, and flexible imaging data analysis environment that leverages software containers. NeuroDesk is an Aussie-based initiative led by Stefan Bowman, and he's over there. And at its core, it is a community-driven initiative with the goal of providing the neuroimaging community with a data analysis platform that enables reproducible research. So how does NeuroDesk work? NeuroDesk hosts containerized software so that you can reliably access a comprehensive range of neuroimaging software. There are over 200 gigabytes of neuroimaging software available via NeuroDesk, and it is delivered on demand in unpacked singularity containers via the CVMFS, which is the CERN-based file system. NeuroDesk has many different interfaces, either a graphical user interface, a GUI, or a command line interface, and it is portable to multiple computing environments. For example, we can have NeuroDesk on our local computer, which is shown up there on macOS. And to do it, you just have to install, for example, NeuroDesk app, which provides both the GUI and the command line interfaces. You can also generate a NeuroDesk instance on cloud resources, or even you can have a NeuroDesk command line interface on a HPC cluster, so you can pull and run any software available via NeuroDesk on demand. And finally, the best of all is that NeuroDesk is completely open source and community driven, which means anyone can contribute with new software to the platform and with tutorials so others can learn how to use new tools and run new pipelines. So if you are willing to give it a go, the first step to learning more about how to use NeuroDesk is to go to our website and go through the different installation options. For example, one of my personal favorites is the NeuroDesk app, as you can use the different interfaces of NeuroDesk within the same application. And here I show how you can install it on a Mac computer, and yeah, it's that easy. Now, once you have a NeuroDesk app installed on your computer, you can explore the different interfaces available. Here I show how you can launch the GUI and how the command line interface, how to use the command line interface. So NeuroDesktop, which is shown there, is the NeuroDesk GUI, which looks like a virtual machine, and it enables different software GUIs. For example, I will show you in a few seconds how you can launch FreeView, which is the FreeSurfer GUI. I'm going through the application menu, and in a few seconds, I will click on FreeView, and there you go. Easy. You can also prototype your analysis via the command line interface. So whenever you feel ready, you can scale your pipelines up by porting your analysis to a HPC cluster, leveraging our command line interface, and it should look the same. So in here, you just have to module load the tool, and I'm module loading FreeSurfer, and that should work the same way in a HPC cluster. You can similarly use this software on a Jupyter notebook using a Jupyter Lab interface, which enables analysis prototyping, documentation, and teaching. And it's also very simple. So I recorded this video a couple of weeks ago. So we were using LMod, but now the new library, or they renamed the library, it's modules. And you, instead of LMod, you just type modules. And you can see we can similarly load FreeSurfer into a Jupyter notebook and run Reconol. So as I mentioned, you can port your analysis to a HPC cluster by leveraging NeuroCommand, which is the NeuroDesk command line interface. And as you can see here, you just have to run a few command lines to set up NeuroCommand and download containers on a HPC cluster. So if you want to find out more, you can scan this QR code with documentation to set that up on your local HPC cluster. So with NeuroDesk, we can prototype our analysis locally, for example, using NeuroDesk app. And then we can port them to a HPC cluster to scale it up using NeuroCommand. And finally, because NeuroDesk is a portable platform, it enables various collaboration models. For example, if you are interested in a centralized collaboration model where multiple collaborators across different institutions have access to centrally located data, one could have a shared NeuroDesk instance with central data access, which enables simplified data management. Alternatively, when working with patient data or when data sharing legislation is stricter, you may need to choose a federated collaboration model in which researchers across institutions collaborate by running the same algorithms and pipelines on their private data, which is stored at their institutions, and that ensures data privacy. And only the metadata or stats will be shared for aggregating results for publication. So if you want to give it a try, you can also check out some tutorials we have available on our website and example notebooks with different neuroimaging pipelines. And of course, if you're also interested in contributing with tutorials and new tools, you can reach out to our team and let us help you help us. And here you can even find a tutorial on how to use a tool that I've developed for predicting retinal topic maps from brain shape, which is already available on NeuroDesk. So because NeuroDesk enables reproducible and portable analysis, it fits perfectly with the open data and open analysis lifecycle. For example, we can easily import data from scanners, open data repository, and cloud storage. Then we can process this data using the rich set of tools available via NeuroDesk. We can also organize our data, document our analysis, and share our code and data with the community, all from NeuroDesk, all within NeuroDesk. So if you are interested to know more about NeuroDesk, you can check out our paper. And please make sure you cite our work if you find it helpful, so you can support us to continue expanding our platform and also improve our platform. But enough of me talking for now. Ashley will take over and will share what Brain Life is about.
Thank you very much. Yeah, so my name is Ashley Stewart, and I'm a research fellow in Stefan Bollmann's NeuroDesk group as well. But I recently did a short-term postdoc in our sister group in Austin, Texas, in the Brain Life group. So I'll talk a little bit about Brain Life, which is another platform for reproducible neuroimaging that leverages containers and then makes that more accessible. So Brain Life is a platform that fills a different purpose than NeuroDesk, groups for different users with different goals. But like NeuroDesk, it enables reproducibility through its use of containers. But it uses what we could call an integrative approach. So it could be described as a free and open source secure, reproducible neuroscience analysis platform. And we'll see a bit what this actually means and what this looks like. So like NeuroDesk, BrainLife is a massively global and open source project, as I said, based in the Pestilli lab in the United States in Austin, Texas. And it has over 4,700 user accounts and 100 monthly users. So it's a large, growing project. And so what BrainLife has really succeeded at is seeing that researchers are burdened by wearing so many different hats. So to run a study, they need to manage tasks with fragmented solutions. So often with no standard way of actually solving them. And this makes science inefficient. And so what BrainLife tries to do is to flip this around and give power to researchers by providing standardized solutions for each of these problems that are integrated into its ecosystem. So while NeuroDesk gives you access to the tools you're familiar with in those desktop and command line environments that aren't going away anytime soon, BrainLife gives you standardized ways of solving each task so that you can focus on the science. And so this integrative infrastructure was developed, as I said, in the Pestilli lab, in no small part by the core developers Bradley Karen, Soichi Hayashi, and Annabel Heinsfeld. So to best understand BrainLife, it's best to check it out. And so we can see here how it brings data to life. So here we have the BrainLife platform. We can go into the portal here if we log into our user, and we have projects. And so we can see all of our data for each project. We can, so this is one project I've selected. We can see the data that's available, grouped by subjects. It's a fairly easy to navigate interface. We can filter the data in different ways. Here we have some white matter tracked data. And it brings out that information that's hiding within that data and then makes it very, very accessible. And then as well as that, we've got lots of different ways of visualizing the data. So there are different data visualization tools that match the data that you're looking at. So these are all running in the browser. You don't have to install anything. And we can, play with and understand and, bring data to life, I guess. So that gives you an idea of what it looks like. But how do you actually use BrainLife for a study? What does that involve? We could split it up into three main steps, where data is at sort of the center of each step, from data import to and standardization, data processing via what they call apps, which are kind of the containerized tools, and then data analysis and publication. So we'll look at each of these, starting with the import and standardization. And so for integrative solutions like BrainLife, to make the most of your data, it needs to understand what it's looking at. And so that's why standardization becomes so important. So BrainLife understands BIDS, the standardized brain imaging data structure. But neuroimaging data is complex, and understanding BIDS is daunting for users that are new to it. And so that's also why within the Pestilli lab, EasyBids was developed. And so this was led by Daniel Levitas and Soichi Hayashi. And EasyBids is, again, a browser-based tool you can access. You can run it locally as well through Docker. And it interactively guides you through this standardization process to BIDS, the brain imaging data structure. And we can see briefly how it works here. So first we find our DICOMs on our system, and we load them. And then once we've loaded them, the next steps of EasyBids take us through confirming what it's identified in the data set. So subjects, extracted demographic information, acquisition information, and other relevant BIDS fields that we must or may provide to finish that conversion to the brain imaging data structure. And so it guides you through this step by step. I won't explain all the details of this, but you can always read more about what it's asking you. So you can also deface your data and anonymize it. And then once you finalize it, we can download it or push it straight into a BrainLife project where then you can start analyzing and running your project. So here we've got download as a zip or send to BrainLife, which we've just clicked on. And after we send it to BrainLife, it's immediately saying, okay, we can create a new project for this BIDS data set that we've just imported, and then it stages it and pulls it so that we can actually start analyzing. So that's all well and good for importing your own imaging data, but how can a platform like BrainLife help when there's a growing wealth of large open data sets, like from OpenNeuro or INDI, for example? So are we meant to manually download these and try to run them through all easy bids? This would be a significant labor. And so this is a really powerful feature in BrainLife. It understands and integrates solutions for these kinds of tasks. If we go into data sets, BrainLife can import data sets using DataLad, giving access to repositories like OpenNeuro. So this is a UCLA data set, and we can just click import, say we want the diffusion data from this open repository on OpenNeuro. We can create a new project for it and give it a name. And then immediately in this archive within the project, we can start to browse demographic information and the data itself in that archive area. So we can see all the subjects that have been imported. We ask for the diffusion images. We can start to visualize and look at them and analyze them as well. And you can see the view is changed depending on what kind of data you're looking at. So this is diffusion data, which has different viewers than, say, the white matter tracks that we looked at before. Okay, so we've imported our data. That's where the next step now comes in, so we can move on to data processing, which is where that containerized apps come in. So we've seen that BrainLife puts data archives and standards at the center there with that import process, but it has another side underpinned by software libraries and compute resources. So how does it actually run these things? And what makes this possible is a technology called BrainLife Amaretti at the center here. And this is a decentralized task scheduler for BrainLife. And it's what connects the data that you've imported in your project with the apps that you want to use and notebooks that you might want to run through to cloud and HPC resources where they will actually run on, like Jetstream, Frontier, Azure, and other compute resources that are available. So this is the architecture, but where is it actually running? Well, as I said, Amaretti is decentralized. So it will send your tasks to run on hardware resources that are available across the world, but there are critical hubs in the US where BrainLife is based, and further compute provided by Azure, AWS, and Google Cloud. BrainLife has access to millions of free compute hours funded by NSF, National Science Foundation, and from donated compute time from any institution that contributes their own HPC resources. So there's a client that you can set up at your if you want to do compute locally, there's actually options to do that. Or if you want to donate compute to BrainLife, that's possible as well, to make the most of hardware that's available. And so on this hardware, we can run a range of familiar containerized neuroimaging software tools on our data. And so what does that look like to run ourselves? So there's several ways that we can run these through BrainLife. But let's just run through one manually across our UCLA project that we just created. So in BrainLife Portal, we'll navigate to apps, the very, very top left, where we can see which apps are available. We'll do some fMRI processing with fMRI prep. And we can see what inputs, standardized inputs and outputs fMRI prep requires. We can see different configuration options we could choose. We can see the different compute resources that this could possibly run on and the maintainers and so on. Going up to the top, we have a README where we can see how to use it, what it's for, again, the authors and other information. We can see recent jobs that have been scheduled, whether they were successful or if they failed and why, whether it was a data problem and so on. And lastly, we can just press execute. So we'll say, well, which project will we run fMRI prep on? Well, we'll run it on UCLA, and we'll make sure we're selecting our T1 from that project and submit. And then it will just start doing the processing across the entire data set. Do I have anything else? Okay. Just move forward. Here we go. And so we can also run apps on individual or subsets of our data. So here we're looking at our data archive for the project. And here we'll choose an example of white matter tracks, one individual data set that we could stage for processing. So we can see the information about it. But up here we can, if we select that data set, we can stage that single example for processing. And that creates a new process that we could queue a pipeline of apps onto. And so here we see one app is available, participant report. So this app list is automatically filtered so that only the apps appropriate for white matter tracks are actually shown. So that really makes so if we look at fMRI data, for example, we have a different set of apps available that we could run on it. So the standardized data types that we're using, we can really see the benefits of that already in how it helps us set up pipelines that actually make sense for the available data. Yeah. So we have Amaretti, as I mentioned, the decentralized task scheduler that allocates our software and data to compute resources. But because apps use these standardized data, we can easily chain together applications for longer and fully reproducible processing pipelines. And so as you do this and create further derived data types in a study, the full provenance of each object is retained. And so that means if you look at any given data object, we can see the history of processing steps, including software versions, and specific configuration options that were applied to it in order to produce it. And so this is becoming critically important for reproducibility in neuroimaging. So that's data processing, and if you're a developer or interested in bringing other apps into BrainLife, there are instructions, a similar container recipe format to NeuroDesk for you to do that, because it's an open source project that relies on community contributions. And so lastly, let's briefly check out how BrainLife can help with data analysis and then publication. So you can publish your work on BrainLife in the publications area of the portal. So on the side there, you can see publications. So here we see multiple publications that we can look at. We can scroll down through them. We can see titles, descriptions, authors. So you could have a browse of that. But let's go into one specific example. So we'll go into the Nigerian brain MRI data set that's on there. So if we click on that, we can see that this data set has a DOI, so that's provided by BrainLife, so the work can be cited, the data can be cited. We see authors, title, abstract. We can see the data that's been available by the project, 51 subjects, which we could download as well. There's a download button there, which will download it in the standardized BIDS format. We can see what pre-processing was applied to it. So here we see some, I think, brain extraction and MRI QC. And then we have an analysis page where we can have a Jupyter notebook that mixes description with code. And so here, this is a Python-based notebook, and it results in an interactive and sort of reproducible paper in a sense. And so, yeah, these publications are really quite powerful, and that's, I guess, a summary of how BrainLife works and brings things together. So I might bring it to Fernanda to wrap up. Yeah, thank you very much.
In sum, we learned today that software containers are standard and isolated units of software, and that they enable reproducible research as they are portable to any operating system and computational environment. And the adoption of containers in science has resulted in this flourishing, in the flourishing of neuroimaging platforms like NeuroDesk and BrainLife, which give the benefits of containers to researchers. And as you can tell, these are complementary platforms, each providing unique benefits. Like NeuroDesk is flexible and portable, while BrainLife provides integrated and standardized solutions. Thank you very much.